Hello, BISC130. This is the beginning of recorded lecture 4.2, continuing on with this genes and proteins chapter. So last time I had introduced the central dogma of molecular biology, and we had talked about transcription. Coming up is translation, but before that, I actually want to outline the three main types of RNA. So yeah, we talked about transcription, how RNA is made, but we need to understand that not all RNA has the same fate. Not all RNA has the same job. There are three different types of RNA, and they do very different things. So one of these is called rRNA, lowercase r before the capital R, capital N, capital A. The little r stands for ribosomal, because this type of RNA forms the ribosomes, as the name implies, which again is this one's kind of unintuitive. You know, RNA is supposed to be translated into protein. It's supposed to be a blueprint. Well, this is kind of an exception to that. rRNA is not a blueprint. It's not a message. It's not instructions. rRNA is a structural component of these ribosomes. It plays a physical enzymatic role in these structures. So, in summary, rRNA, ribosomal RNA, a structural component of ribosomes. We'll see ribosomes in action soon enough, but yeah, this type of RNA is a structural component of these things. Another atypical, but very important type of RNA is tRNA, a little lowercase t, RNA. The T stands for transfer. And I'm honestly not going to say much about these here, just kind of introducing this term. We're going to see these in action when we talk about translation. And once again, these are not a message, they're not a blueprint, these are playing a structural role that we will see later in this chapter. So tRNA, the T stands for transfer. These things play a structural role in the process of translation. And finally, it's the one I've been saying the others are not. Our third type of RNA is mRNA. It is messenger RNA. It is the blueprint. It is an instruction. mRNA is not playing some structural role in a process. It's just the information. It's just the message. So mRNA, messenger RNA, its information directs protein synthesis. Just like a blueprint directs the building of the house, the information, the code within this messenger RNA is going to tell the cell how the protein is supposed to be made. It's the message. The other ones are structural RNAs. Okay, so now we can move to translation. Well, not quite yet. Before we talk about the process of translation, we have to talk about something called the genetic code. So, little background. How many different nucleotides are there? Hopefully you know this one off the top of your head, the number of different building blocks that are used to build DNA. It's 4. Adenine, guanine, thymine, and cytosine. Not uracil, that one's used in RNA, but yeah, there are only 4 different nucleotides that are used to build DNA. What about RNA? How many building blocks does RNA have? Well, it's also 4. It's adenine, guanine, cytosine, and instead of thymine, we've got uracil. So there are 4 different building blocks in RNA. What about amino acids? This one's a little trickier. We haven't talked about amino acids in a while, but if you've got a sharp memory, you may remember a table like this from several chapters ago, and you may remember, even if you didn't memorize the names of all of these things, that there were 20. So this is a pretty big disparity. When we were doing transcription and we were going from a piece of DNA to a piece of RNA, it was a pretty easy 1-to-1, one nucleotide in DNA corresponded to one nucleotide in RNA. That worked out pretty easily because, yeah, they have the same number of letters in these 2 alphabets, if you want to think about it like that. But if we try to do 1-to-1, if we try to have one, as we move to translation, if we try to have one RNA nucleotide code for one amino acid, clearly that's not going to be enough. We're going to be missing 16 codes here. If the genetic code was 1-to-1, That's not enough. 4 is nucleotides, 4 letters is not enough to somehow encode 20 different amino acids. So the genetic code is not going to be 1-to-1. What about 2-to-1, though? So what if we have 2 nucleotides encode 1 amino acid? So by 2 nucleotides now, I'm talking about a code, something like AA, or GC, or UU, or UC, or CU, or all of these. I did not list all the possible combinations of 2. If you're a math person, though, you'll be able to understand that the number of possible combinations of 2 nucleotides is 4 to the second power, because there are 4 possibilities for each position, and there are 2 positions. If you don't follow the math, don't worry about it. But there are 16 possible combinations of 2 nucleotides, which is cool, but it's still not good enough. Again, we need 20 for our 20 amino acids. So the genetic code, whatever it is not 2-to-1. What about 3-to-1? Well, you can follow the same kind of math. If 3 nucleotides encode a single amino acid, don't know why I said 1 up here, but single down here, whatever, the number of possible combinations of 3 nucleotides is 4 to the third power, which means 64. There are 64 possible combinations of, CCU and UGA and GGU and things like that, which is more than enough for our 20 amino acids. So I could have just told you that the genetic code is based on sets of 3. I went through this so we could understand why. It can't be 1-to-1. 2-to-1's not good enough. 3 is the simplest code that gives us what we need. So the take home here, the genetic code is going to be based on sets of 3 nucleotides. There's a very important term that refers to these sets of 3. These things are called codons. I'm going to move to a table now that you do not need to memorize, but it's important to be able to look at and interpret. Here's our genetic code. These are the 64 different possible combinations of 3 nucleotides, RNA nucleotides, ACU, AAU, GAU, GGU. Yeah, it's all 64 of them in this big old table. Now, there are a couple of things that you may notice about this table. One of these should be the redundancy. So we have 20 amino acids and 60 codons. That is, as I've noted, more than enough. So instead of having codons that just do nothing, we have redundancy. So if we look at this, if we want to encode the amino acid proline, for example, CCU codes for proline, but so does CCC, and so does CCA, and so does CCG. In fact, almost all of these amino acids are encoded by 2, 4, sometimes even 6 different codons. The term for this redundancy, and this is kind of a strange term, but I didn't make this up. This is the term that's used in the world of code making. The term for this redundancy is to say that the genetic code is degenerate. And again, that doesn't imply it has bad morals or something like that. The degeneracy of the genetic code means that there's redundancy in the genetic code. Most amino acids are encoded by multiple codons. That's what this means. The other thing that you should hopefully notice if you pause and stare at this for a second is most of these codons are encoding amino acids. That's what these 3-letter all 20 of these amino acids have a cute little 3-letter code. They also have cute little 1-letter codes. If you take biochemistry, you'll probably have to memorize all of them, but for here, don't even begin to worry about it. But yeah, that's what these things are: proline, threonine, leucine, isoleucine. That there are a few codons here that seem a little abnormal. There are, in fact, 2 different types of what we call special codons. One of these is the one in red right here. The AUG red codon right here is called the start codon. So this codon, there's only one start codon. It is a type of special codon, and there's only one of them. The start codon signals the start of the protein. We're going to see this in action when we finally start talking about translation. But yeah, that's the job of the start codon. It says translation, start here. This is the beginning of the protein. It's going to put in the first amino acid. Importantly, it does two things, actually. Not only does it say start here, it also says methionine. That's one of those 20 amino acids. So the start codon also encodes the methionine amino acid. The other type of special codon is over here. It's called the stop codon. It's actually not the stop codon, it's the stop codons, because there are 3 of these. All 3 of these codons do the same thing. They say translation, stop, end of the line, end of the protein. In contrast to the start codon, which encoded an amino acid, the stop codons do not encode an amino acid, they just say stop. So there are 3 different stop codons. Again, you don't need to memorize their sequences, but you do need to know that there are 3 of them. There are 3 different stop codons. They signal for the end of translation, and they do not encode an amino acid. One other important thing about this code. So this code that we've been looking at here, I should more appropriately, label this. This is the human genetic code we've been looking at. So this is how our bodies, our cells, go through the process of translation. But let me switch to, I think I have cat coming up next. So for keen-eyed viewers, keep an eye out for this. I'm about to switch over. This is a spot the difference moment. So this is human. I'm going to switch to cat now. Did you see it? Let me do another one. Coming up next is earthworm. Coming up next is oak tree. Coming up next is E. coli, bacterium. I'm screwing with you. They're all the same. The genetic code is almost completely universal among all living things. The 64 codons and what they encode did not change at all in any of these examples. There are a couple of weird primitive bacteria that have a couple of codons that are different, but other than those weird exceptions, even then it's only small differences. It's all universal among, certainly all eukaryotes, certainly all animals and mammals. It's almost completely universal. The genetic code is almost completely universal, is the blanket statement to make concerning this. There's an interesting example or demonstration of this from the textbook, showing this glowing kitten. So, well, this is cool, but there's a reason for this. This kitten is glowing green because it is transcribing and translating a protein. This protein is called green fluorescent protein, and it fluoresces green. It does exactly what the name says. This is not a protein that kitten normally has. It's actually a protein that's found in nature in some species of jellyfish. But because the jellyfish genetic code is exactly the same as the cat genetic code, you can take the gene for this protein from one organism, put it into a different organism, it's going to get transcribed and translated faithfully, and they're going to make the same protein that the jellyfish make. So yeah, this is just demonstrating the universality of the genetic code. And if you're curious, this is not just mad science to make glowing cats. This is proof of concept for being able to introduce genes into higher vertebrates like mammals. This is proof of concept for gene therapy to try to cure genetic disorders in humans. If we can introduce a gene into a cat, it's green just because it's easy to see whether it's succeeded or not. Maybe we can fix genetic issues in humans. So yeah, looks adorable, but there's a reason behind this madness. And I'm just using this experiment for you to illustrate this universality. Okay, so now that we know a bit about the genetic code, we can finally start talking about translation. So, translation is carried out by structures called ribosomes. These are not just structures, these are actually enzymes. They carry out enzymatic reactions. One of the few enzymes that doesn't end in ACE, but it's kind of a special one because they're massive, massive structures. So translation carried out by ribosomes in 3 phases. The first phase is called initiation. This begins when we have the small subunit of the ribosome. Oh, by the way, yeah, the ribosome consists of 2 subunits, a small one and a big one, and they come together. So initiation starts with the small part of the ribosome binding to our messenger RNA. Small ribosomal subunit binds to mRNA. From there, a tRNA, I told you that I would come to these, a tRNA, which specifically holds the methionine amino acid, binds to the start codon. I know I told you didn't have to memorize this, and that's still true, but if we want to double check this, AUG, yep, AUG, there's our start codon, so that's exactly what I said it was. This special tRNA is called the initiator tRNA, carries methionine, binds to this start codon. From there, the large ribosomal subunit is going to bind. Sorry, I blocked out some text that's confusing at the moment. The large ribosomal subunit is going to bind, and now we're ready to start building protein. So after this initiation of everything sort of coming together, we move to our second phase called elongation. Now, to talk about elongation, we kind of have to go back to these tRNAs and talk a little bit more about their actual structure. tRNAs have 2 important bits to them. One end is the end that holds the amino acid. The other end, down here, is called the anti-codon. So tRNAs have an amino acid attached at one end and an anti-codon at the other. The anti-codon is exactly what it sounds like. It's like the reverse of a codon. Anticodons are complementary to codons that are present in the mRNA. So if we go back to this initiator tRNA, just to use this as an example, the codon was AUG. That's the start codon. Therefore, if we have a tRNA, if we want it to bind to this start codon, this initiator tRNA must have an anticodon complementary to the start codon, which would be UAC. It's because A is complementary to U, because again, we're talking about RNAs here. There's no thymine, there's uracil. U is complementary to A, G is complementary to C. Bam, there we go. That's why this initiator tRNA binds the way it does. Its anticodon matches up with the start codon. That's why it comes into place. Because there are tons of different tRNAs floating around in the cell. Only the ones that match up with the exposed codon are the ones that are going to enter the ribosome. So actually, let's move to the next step here. We see our next codon is UUC. So UUC is complementary to AAG. So there should be a tRNA floating around with AAG. And which tRNA would it have? Well, we're going to have to go back to the genetic code, which again, we don't memorize. What is UUC supposed to encode? UUC is supposed to encode phenylalanine. So that means in the cell, there should be a tRNA floating around with phenylalanine attached at one end and an AAG anticodon. Skip to this. There we go. Phenylalanine, AAG. This of all the tRNAs in the cell is going to enter the ribosome because it's the one tRNA that fits correctly. Its anticodon matches up with this exposed codon in the ribosome. So, to summarize in text, tRNAs enter the ribosome one at a time during elongation according to the mRNA sequence. If we want to do another step of this, moving next, looks like our next exposed codon is CGA. Going to the genetic code, let's see, CGA. What is CGA supposed to encode? There we go, CGA, arginine. So there is going to be a tRNA floating around with arginine attached to one end and an anticodon of GCU. Let's double check this as we go back. GCU, arginine. This is the next tRNA that's going to enter the ribosome. And this is where my figure ends, but the cell is going to go through this exercise again and again, because there's going to be another codon and another codon and another codon and another codon. They're all going to enter the ribosome one at a time according to the mRNA sequence, one tRNA at a time. Now, we kind of skipped over an important part here. Enter one at a time according to the sequence. We kind of skipped over an important part here, the attachment of this first amino acid to the second amino acid. Remember, in order to build protein, we have to connect amino acids to one another. We have to create, if you remember this term from several chapters ago, a peptide bond. So as part of elongation, the tRNA's amino acid is passed to the next tRNA's amino acid. Peptide bond created, and yes, this costs energy. Building protein costs a lot of energy. Every single amino acid that you attach to the next one is going to take energy to form this kind of bond. So we're going to pass the amino acid to the next amino acid. From there, the empty tRNA, this one has lost its amino acid, it gave its methionine to the next one. This empty tRNA is going to exit the ribosome, and the next tRNA is going to come in. So empty tRNA exits, sorry for that typo, exits the ribosome as the mRNA slides along, and the next tRNA comes in, etc. Again, this is just showing the first three, but there will be a fourth, a fifth, a sixth, and so on with every tRNA coming in according to the mRNA sequence. mRNA sequence, anticodon carrying the appropriate amino acid. This is going to continue on for, depends on how long the protein is. Most proteins are dozens, hundreds of amino acids long. This will keep happening until we reach our third phase called termination. During termination, there is a stop codon that is exposed in the ribosome, one of these three. There is no tRNA floating around that corresponds to the stop codon. Instead, there is a protein called release factor that recognizes the stop codon. This is not a tRNA. It looks like a tRNA. It has the same shape as a tRNA. It would have to. It's fitting into the little pocket that tRNAs normally occupy. But this is a protein. The release factor protein, upon recognizing a stop codon and entering the ribosome, is going to cause the whole thing to fall apart. The two halves of the ribosome fall apart. The mRNA is released, and the protein is released as well. This string of amino acids, it's going to fold up into its complicated three-dimensional structure if it hasn't started doing that already. And so, this termination happens because a stop codon pops up. So to summarize, a stop codon in the mRNA causes release factor protein to come into the ribosome. The two ribosomal subunits dissociate or come apart. The protein is released, and the mRNA is released, And importantly, so yeah, this mRNA, if you think about what happened to this mRNA throughout this entire process, the mRNA is not altered by this at all. It's just sort of looked at. It's just sort of read, and then it's going to slide down. And the next tRNA comes in, but nothing is permanently happening to this mRNA. And so once this mRNA is released, there's absolutely nothing stopping that mRNA from just going right back to the beginning and becoming translated a second time, a third time, a fourth time. mRNA doesn't have the best shelf life, so it doesn't last forever. Eventually, it'll start to break apart, and it'll be scrapped for energy and spare nucleotides. But yeah, that mRNA is going to be translated again after it has been released. So importantly, I know it's confusing that the 3 stages of transcription were initiation, elongation, and termination, which are the same stages as translation. But it really is important to remember the difference between transcription and translation and what is actually doing what. Importantly, translation starts at the start codon and ends at a stop codon. Transcription, if I can go back to this as just a means of compare and contrast, transcription started at the promoter and ended with either Rho factor or an RNA hairpin. So even though they both have beginning, middle, and end phases, it's very important to not get them mixed up and remember what starts and stops and does transcription. Remember, RNA polymerase does transcription, and not get that mixed up with the things that start, stop, and perform translation. Ribosome does translation, starts with a start codon, and ends with a stop codon. That's all just review. So, termination, there's my piece for translation. Now, once again, eukaryotes v. prokaryotes. So how are they different when it comes to translation? Well, there's really one kind of minor difference that kind of has a big impact, and that has to do with where all of this is happening. So there's a lot going on here. Let's look at this left half first. So this is a prokaryotic cell. And if you'll recall, prokaryotic cells do not have a nucleus. So we're watching the process of transcription right here. This is our big molecule of DNA, again, a circular DNA chromosome. And here's the creation of mRNA from that DNA. Transcription is occurring right here. Because this is just floating around in the cytoplasm, the cell actually doesn't have to wait for transcription to finish before ribosomes can come in and start translating that mRNA and building protein. So these two steps are happening simultaneously. Transcription is still ongoing. It hasn't even finished reading the DNA yet, and yet translation has already begun. So in prokaryotes, we have simultaneous transcription and translation. However, we don't in eukaryotes. Eukaryotes do have a nucleus. The nucleus is where transcription happens, and the cytoplasm, or the rough ER, is where translation happens. So we can't do them at the same time. In a eukaryote, we have to fully transcribe this mRNA. We've got to do our processing, our splicing, our additions for stability. Then it gets exported from the nucleus to where it needs to go. Then we can do translation on this mRNA. So yeah, simultaneous in prokaryotes, but in eukaryotes, transcriptions in the nucleus, translations in the cytoplasm. So again, that's the biggest difference between these two. Everything else that I had said about translation applies to both prokaryotes and eukaryotes. Now, finally, we're coming back to this. So this was in the last chapter. It's technically a chapter 14 topic. I told you we would come back to it later. Well, we're coming back to it now to talk about the different types of mutations. The first category of mutation I want to talk about is called point mutations. And you can see there are three of these four. Three different types of mutations fall under this category of point mutations. Point mutations are changes in a single base pair. So whatever has changed in the sequence is just a single change, just one base pair has changed. There are three different types of point mutations because it kind of depends on how it has been changed. Let's take a look at the first type of point mutation called a silent mutation. So you can see the mutation here. It's within this codon GTA. It looks like the A has been mutated. Again, a mutation is a stable change in the DNA sequence. This GTA, the A has been changed to a T. So instead of being the GTA codon, it is the GTT codon. Now that we've learned about the genetic code, we can investigate what effect that's going to have on the protein. Well, sorry if this is confusing. This is showing mutations not in the RNA strand, but in the coding strand of the DNA. So remember, the coding strand and the RNA have the same sequence, it's just U's instead of T's. So we won't be looking for GTA in the genetic code. We will be looking for GUA and comparing that to GUU. Just a call back to those different strands and a template and coding and all of that stuff. So GUA encodes valine, whereas GUU encodes valine. Oh, so remember the genetic code is redundant. So sometimes you can have a mutation that actually doesn't change the amino acid sequence at all. The DNA sequence has been changed. This is a mutation, but the protein that comes as a result of this is exactly the same. It's not going to change the protein sequence. That's why this is called a silent mutation. No effect on the amino acid sequence. The new codon encodes the same amino acid that the old codon did. So that's a silent mutation, a type of point mutation. Next up, we have something called a missense mutation. In this example, we're looking at this codon here, CCC, and the mutation is right there at the first position. Instead of a C, it's an A. So the CCC codon has been changed into an ACC codon. If we consult the genetic code, there's our CCC, which encodes the amino acid proline, and there's ACC, which encodes something different. Again, this is not silent mutation. We're actually changing the sequence of this protein. Instead of a proline amino acid, there is going to be a threonine amino acid in this position. So in a missense mutation, the amino acid, one of the amino acids of the protein, is actually changed. Now, this might not seem like a big deal. Again, there are dozens, hundreds of amino acids in a protein. Is changing one of them really going to affect the protein's function? Well, it absolutely can. Sickle cell anemia is a genetic disease, for example, that is caused by a single amino acid changed from one to another. So missense mutations, one little amino acid change can have a very profound effect on the functionality of a cell. Third type of point mutation is called a nonsense mutation. Looks like we're over here now. TAC is now TAG. Remember, this is a sequence in the coding strand of the DNA, so we won't be looking for TAC in our genetic code table. We will be looking for UAC. UAC encodes tyrosine. We've changed UAC to UAG, and it looks like UAG encodes, a stop codon. So instead of just swapping one amino acid for a different amino acid, in this type of mutation, a nonsense mutation, we cause an amino acid encoding codon to become a stop codon. So protein's just going to end here. Doesn't matter whether it was near the beginning or in the middle or wherever it is, you're just going to stop prematurely and you're going to have a protein that is shorter than it is supposed to be. So nonsense mutation, this is where an amino acid encoding codon is changed to a stop codon. And yeah, this can also have catastrophic effects on the functionality of that protein. Cystic fibrosis, for example, is caused by a nonsense mutation. Now, all three of these were examples of point mutations. One nucleotide was changed to another nucleotide. The other category of mutations is called frame shift mutations. In a frame shift mutation, we are not changing one nucleotide to another, we are adding or removing entire nucleotides from this sequence. Here's an example of a frame shift. So we've got AGC, GTA, CCC. In this example, the TA has been removed. Then instead of GTA CCC, it's just G CCC TAC and the rest of the sequence. What this does, I'm not even going to track this. It screws everything up. Every single codon after this point is going to be messed up, is not going to be read correctly. Again, the reading frame of codons is sets of 3. If you add or remove 1 or 2 nucleotides, it messes up the frame of reading. So my example of what this would look like, here's a normal looking sentence. The sentence reads, would this make sense if I shifted each space? And here I'm going to introduce a shift here in the reading frame, just moving that little space over 1 point. Now the sentence reads, would to hism aches ense fees hif td ox pace? And obviously resulting in total nonsense. Don't get it mixed up with actual nonsense. That was a different type of thing, resulting in everything being wrong. So a frame shift mutation, like I said, a few nucleotides are inserted or removed. If that is not a multiple of 3, if it's a multiple of 3, you're just going to get a new amino acid or you're going to lose an entire amino acid. But if this addition or removal is not a multiple of 3, it's going to completely change the further amino acid sequence in this protein. Again, this is only showing a couple downstream. Proline, tyrosine becomes leucine, but yeah, it's going to mess up everything that comes after that. So again, to summarize, we had point mutations, which came in 3 varieties: silent, missense, and nonsense. And then we have this frame shift mutation. And hopefully you understand why I moved this to chapter 15. I really don't understand how they expect students to understand these before we've introduced the genetic code and how this works. So yeah, that's why I moved it over to chapter 15. I think you need to understand this first before you can understand the effects of these mutations. But anyway, that's the mutation stuff. Now, I do briefly want to start in on the next chapter within this recorded lecture. I know it's kind of long already, but I'm just briefly beginning on this. So the textbook calls this chapter 16 gene expression. I'm going to just quickly change the name of this to call this chapter regulating gene expression. This just makes more sense in my opinion. So what is gene expression? Well, the phrase gene expression refers to everything that we've just talked about in the last chapter. The process of a gene in the DNA being transcribed into mRNA, being translated into protein, and then those proteins doing stuff in the cell, all of this business. We just refer to that as gene expression. And if a gene is expressed and it's going through transcription and translation and its proteins are doing stuff, that gene is said to be on. If a gene is not expressed, and for whatever reason, at whatever point, it is not going through this entire process, that gene is said to be off. Now, this leads to kind of an interesting question. Why would we need to turn genes off? What's the point of having these instructions if you're not going to use them? Why would you waste space in the chromosome on something that you're not going to transcribe and translate? Well, an important thing to point out here in the overall process of gene expression is we've got a lot of genes, but we don't need all of them at all the time. Not all genes need to be expressed at all times. A good justification for this statement comes when we look just at our own bodies. So here's a slide I stole from BISC132, but yeah, these are some of the internal organs of the human body, and every single one of the cells in all of these organs has the same chromosomes. They have the same sets of genes. They have the same instructions. Your neurons have the instructions for how to build muscle proteins. Do your brain cells need to be constructing muscle proteins? No. And your muscles have all the genes required for making liver proteins and, metabolizing things. Do your muscle cells need to be expressing those genes? Absolutely not. So even though we have, all these cells have the exact same genetic instructions, most of these genes are going to be turned off in most of these cells for them to do their jobs. In summary, and sorry, this is kind of an awkward sentence. It's the best I could come up with. Different cell types express different genes in order to fulfill different functions. That's why we don't need everything on all the time. We have different cells with different sets of genes on and off because they're doing different jobs and need different genes. That's true for multicellular organisms. So for multicellular organisms, that statement is true. But for every organism, this is also an answer to the question of why we have genes that we're not going to use in order to respond to the environment. Some genes are only necessary under certain circumstances, and so we don't need them to be on all the time. We want to have them on hand if needed, but we need to be able to switch them on or off in order to respond to the world changing around us. That's why I wanted to call this chapter Regulating Gene Expression, because this chapter is not just the process of gene expression. We kind of did that in the last chapter. This process is about how we control this, or this chapter is about how we control gene expression, how we switch things on or off. Now, there are different ways to switch things on and off. In prokaryotes, most of this control is right here at the beginning, at the transcriptional stage. In prokaryotes, if you want to control whether a gene is expressed or not, usually it's right here at the beginning, controlling whether the gene is transcribed into mRNA or not. To do this, things called transcription factors are used. Couple of key terms here. Transcription factor is defined in the key terms as a protein that binds to DNA and influences transcription of a gene or genes. In other words, a transcription factor is a protein that controls this switch. There are two different flavors of transcription factors, ones that turn things on and ones that turn things off. Based on the names, you could maybe guess which one is which, but they're defined in the key terms if you want that spelled out for you. Repressors are a type of transcript. Let me read the key terms. Repressor a transcription factor that prevents or reduces levels of transcription. And activators are, to read from the key terms, a transcription factor that increases levels of transcription. So yeah, these are types of transcription factors, ones that switch off, ones that switch on. These can be used by prokaryotes to regulate transcription. Eukaryotes do use transcription factors. We do have repressors, we do have activators, but in addition to that, we also control stuff everywhere else. We also control at the translational stage, regulating whether the mRNA is translated or not. And we control things at this stage as well. We've already made the protein. We're going to control whether it does stuff or doesn't do stuff. So prokaryotes mostly regulate things transcriptionally. Eukaryotes regulate at every stage. Now, if this sounds a little abstract, like what does this look like? How do you switch on? How does an off switch work? That's just fine, because what's going to come next in this chapter is an example of each of these. So again, I said I was just briefly getting started on this regulating gene expression chapter. In the next recorded lecture, we'll pick up here, and I will give you an extended example of transcription regulation involving both repressors and activators. So yeah, just introducing the terms now. We'll see more of it next time. So that's it for recorded lecture 4.2. More gene expression regulation in the next one.